The following scenario is hypothetical and presented for educational and entertainment purposes. It started, as many scientific stories do, with a creature so small that most humans would never notice it. A microscopic animal drifting quietly through a drop of water on moss, living its entire life between grains of dust. Scientists call it a tardigrade. Most people call it a water bear. Both names are accurate. Neither prepares you for what witnesses claim to have seen. Because according to a strange set of archived reports, some scientific, some governmental, some dismissed as nonsense, this nearly indestructible organism may have been involved in an experiment that went terribly wrong. An experiment so strange that the official records were sealed for half a century. And when they were finally reopened in 2026, the story that emerged was unsettling. To understand why, we first need to talk about the creature itself. The tardigrade is one of the most extraordinary organisms ever discovered on Earth. First identified in 1773 by German zoologist Johann August Ephraim Goes, it measures less than 1 millimeter long and looks like a chubby, 8-legged bear wearing a badly designed spacesuit. It waddles slowly, eats algae or bacteria, and spends most of its life minding its microscopic business. In other words, it is the least threatening animal imaginable. And yet, biologically speaking, it is a nightmare for the laws of nature. Tardigrades can survive temperatures close to absolute zero and heat above 150 degrees Celsius. They tolerate radiation doses thousands of times higher than what would kill a human. They can endure pressures deeper than the ocean trenches and survive the vacuum of space. In fact, in 2007, European scientists sent thousands of tardigrades into orbit aboard the Photon M3 spacecraft. When the capsule returned to Earth, many of them simply woke up. No dramatic recovery, no medical treatment. They just continued living as if space had been a slightly inconvenient vacation. Scientists believe this is possible because tardigrades enter a state called cryptobiosis. When conditions become hostile, too dry, too cold, too hot, the animal dehydrates itself until only about 3% of its body water remains. Its metabolism slows almost to zero. It curls into a shriveled shape known as a ton and waits. Not for minutes, not for days, sometimes for decades. When water returns, the tardigrade rehydrates and resumes life as if time had politely paused. Now imagine a creature that can effectively stop time for itself. A biological pause button. A microscopic astronaut capable of surviving the vacuum of space. Scientists were fascinated. Military researchers, according to some documents, were even more interested. Because if an organism can survive radiation, pressure, dehydration, freezing, and vacuum, it might hold secrets valuable for long-term space travel, cryogenic preservation, or even biological warfare resistance. Which brings us to a curious experiment recorded in a small research facility in Eastern Europe in 1974. The official summary of the experiment is short, suspiciously short. A group of biologists were attempting to study how tardigrades behaved after repeated cycles of cryptobiosis and radiation exposure. Their hypothesis was simple. Perhaps the animals had limits that had not yet been discovered. At first, everything proceeded normally. The tardigrades were dried, irradiated, revived, and observed, then dried again, then revived again. But sometime during the 8th cycle, one of the lab assistants wrote a note in the observation log. It was not a scientific note. It was a sentence written in hurried handwriting. Specimen cluster moving before rehydration. Which is impossible. A tardigrade in cryptobiosis has virtually no metabolic activity. Without water, it should be biologically frozen in time. Yet according to the next line in the log, 3 researchers reported the same observation. The dried specimens appeared to twitch. At first, they assumed it was vibration from nearby equipment. The lab sat next to a mechanical pump system that often rattled the tables. But the movement happened again. And again, still dry, still supposedly dormant. The supervising scientists dismissed the observations as optical error. The experiment continued. That decision, according to later testimonies, would become the first mistake. Because 2 nights later, a security guard reported something unusual in the observation room. Not a broken container, not an alarm, just a strange smear on the inside of the glass chamber where the tardigrade cultures were stored. A smear that, under the microscope, looked disturbingly like movement trails. And according to the guard, the chamber had not been opened, at least not by any human. The smear should not have existed. That was the first conclusion written in the internal memo recovered decades later. The chamber containing the tardigrade cultures had been sealed, cataloged, and placed under a controlled humidity lock to maintain the animals in a cryptobiotic state. No water, no oxygen exchange, no nutrients. In theory, the specimens inside were biologically paused, like tiny living statues. And yet the microscope slides told a different story. When technician Elena Markovich first examined the smear under magnification, she expected contamination, maybe fungal spores, maybe residue from a careless pipette. Laboratories are messy places, after all. Science often advances through small disasters and creative excuses. But what she saw instead made her call in 2 senior researchers immediately. The smear wasn't random, it had structure. Tardigrades, even under a microscope, leave distinctive movement patterns when they crawl. Their 8 stubby legs grip surfaces with slow, deliberate steps. Normally, those patterns appear only when the animal is fully hydrated and active. Yet the trails inside the sealed chamber looked exactly like that. Tiny step-like impressions, multiple overlapping tracks, as if several microscopic creatures had wandered around inside the container. There was only one problem. The chamber still had 0 humidity. The hygrometer needle hadn't moved. In simple terms, the animals should still have been dry. Scientifically speaking, a dehydrated tardigrade cannot walk. In cryptobiosis, its cellular structures are locked in place by special proteins and sugars that stabilize membranes and DNA. The animal is essentially a biological time capsule. Without water to restore its molecular flexibility, movement should be impossible. And yet the tracks were there. The lab staff debated the possibilities for nearly 2 hours. Equipment malfunction, humidity sensor failure, air leak, static artifact on the slide. Each theory sounded reasonable until they tested it. The chamber seals were intact, the sensors worked perfectly, the internal humidity level remained bone dry. Finally, the supervising scientist, Dr. Pavel Orlov, ordered something very simple. Open the chamber. Not immediately, though. First, they documented everything. Photographs were taken through the microscope, measurements recorded, samples logged. Scientists may panic internally, but paperwork always comes first. Only after the records were complete did they break the seal. When the lid lifted, nothing dramatic happened. No explosion of movement. No alien horror. Just a tray of dry moss particles and several dozen tiny tardigrade tons shriveled into their familiar barrel shape. They looked exactly like every other dehydrated specimen ever observed. For a moment, the tension dissolved into embarrassment. Someone joked that the tardigrades must have taken a midnight stroll and returned before anyone noticed. Another suggested the tracks might have formed before dehydration. It was the kind of laughter people use when they are relieved to be wrong. But then, Elena adjusted the microscope again, and the room went silent. One of the specimens wasn't shaped correctly. A normal tardigrade ton is symmetrical, like a tiny wrinkled capsule. Legs tucked in, body contracted, metabolism essentially off. But this one appeared stretched, slightly elongated, as if it had partially attempted to unfold. Dr. Orlov leaned closer to the eyepiece. "Hydrate one," he said. The technician added a microscopic droplet of distilled water. Anyone familiar with tardigrades knows what usually happens next. After rehydration, the animal slowly expands. Over several minutes, its limbs extend, its body plumps up, and eventually it begins crawling again like a microscopic bear waking from hibernation. But this one did something different. The expansion happened almost instantly. Within seconds, the body inflated and the legs unfolded. The creature twitched violently like a computer rebooting too fast. Then it started moving. Not slowly, not lazily, but fast. Much faster than a tardigrade should be capable of crawling. Across the slide it went, bumping into the glass edge, turning sharply, then racing along the surface again in jerky bursts. The movement was so sudden that Elena actually pulled her eye away from the microscope. "What is it doing?" someone asked. No one answered, because Dr. Orlov was still watching. And what he saw next would become the first moment later investigators described as the beginning of the nightmare. The tardigrade stopped moving, not randomly. It stopped directly beside one of the microscopic trail marks that had been photographed earlier. The animal remained still for several seconds, its tiny claws touching the track. Then slowly, almost deliberately, it began to follow the exact same path. Step, step, step. Every movement aligned perfectly with the earlier trail pattern, which meant something deeply uncomfortable. Those tracks had not been random. They had been made by this exact animal. Before the chamber was opened, before any water had been added, before the creature should have been capable of moving at all. And if the records were correct, it had done that while completely dry. For a long moment, no one in the room spoke. Then Dr. Orlov said something that would later appear underlined 3 times in the archived report. "Run the radiation scanner." Because if a tardigrade could somehow move during cryptobiosis, there were only 2 explanations. Either everything scientists believed about the limits of life was wrong, or something inside that chamber had changed. The scanner results arrived 5 minutes later, and that was when the experiment stopped being strange and started becoming dangerous. Radiation scanners are not dramatic machines. They do not scream like movie alarms or flash red warning lights. In most laboratories, they simply produce numbers. Calm, indifferent numbers that either reassure scientists or ruin their day. On that evening in 1974, the numbers did the second thing. The tardigrade sample on Elena's slide showed radiation levels nearly 12 times higher than the controlled dose used during the previous experimental cycle. At first glance, this sounded impossible. Radiation does not simply appear inside sealed chambers. It requires a source, radioactive material, a malfunctioning emitter, or contamination from outside. None of those explanations fit. The irradiation device used in the experiment had been powered down for 16 hours. Its shielding was intact. The room monitors showed normal background radiation levels. Yet, the scanner reading on the specimen remained stubbornly elevated. Drs. Orlov checked the instrument calibration. Correct. He scanned the empty slide surface. Normal. He scanned another tardigrade ton from the same container. Normal again. Only the strange specimen, the one that had unfolded too quickly and sprinted across the glass, produced the abnormal reading. Now, to a biologist, radiation tolerance in tardigrades is not new. These creatures possess extraordinary DNA repair mechanisms. Their cells produce protective proteins, including one famously nicknamed DSUP, short for damage suppressor. This protein shields genetic material from radiation-induced fragmentation. In practical terms, tardigrades can survive doses that would shred human DNA into molecular confetti. But survival is not the same as absorption. The scanner suggested the specimen itself had become a radiation source, or at least was retaining far more energy than expected. Dr. Orlov ordered a second hydration test. Another ton from the chamber was placed onto a slide and exposed to water. This time, the lab staff watched through a projection microscope so everyone could see the process simultaneously. The tardigrade revived normally. Slow expansion, gentle leg movement, the familiar lazy crawl that looks like a microscopic gummy bear learning to walk. The room relaxed slightly. Then the first specimen, the fast one, moved again. It had been resting near the edge of the slide during the second test. Without warning, it accelerated toward the newly revived tardigrade. Under magnification, the moment looks strangely dramatic. 8 legs pumping, body lunging forward. Before anyone could react, the specimen collided with the second tardigrade. For half a second, the two animals simply touched. Then the second one convulsed. Its legs stiffened, its body twisted, and it collapsed into a rigid shape. Elena instinctively adjusted the focus. "Is it dead?" someone whispered. But Dr. Orlov was already shaking his head. No, because the scanner reading had changed again. The radiation level of the second specimen had begun rising, slowly at first, then steadily. If you were to describe the scene purely scientifically, it would sound almost mundane. Two microscopic animals interacting on a slide. No dramatic music, no horror visuals, just biological data quietly becoming more confusing. But to the researchers watching, the implications were deeply unsettling. The radiation exposure from the earlier experiment might have altered something fundamental in the tardigrade's physiology. Perhaps its DNA repair system had overreacted. Perhaps cellular proteins had mutated into a new protective mechanism. Or perhaps, and this possibility appeared later in the classified notes, the organism had begun transferring energy between individuals. That would be unprecedented. Life forms exchange chemicals, nutrients, even genetic material in certain conditions. But radiation transfer through direct contact, that would suggest a completely unknown biological process. Dr. Orlov immediately ordered isolation protocols. The fast specimen was moved to a separate sealed slide chamber. The second tardigrade was monitored continuously. Within 10 minutes, it recovered movement, but its behavior was no longer normal. Instead of wandering randomly, it followed the first specimen's trail patterns almost perfectly, just as the original animal had done earlier. The lab now had two specimens behaving identically, two specimens showing elevated radiation levels, two specimens capable of movement patterns that seemed learned. That was when Elena said something that later investigators would quote repeatedly. "This looks like spreading." No one liked that word. Not in a laboratory. Not when dealing with organisms that could survive almost any environment known to science. Dr. Orlov closed the observation log and issued the order that would trigger the first major climax of the entire incident. Terminate both specimens. The procedure should have been simple. A high-intensity heat pulse would instantly destroy organisms this small. It was standard laboratory sterilization. The heating element activated, temperature climbed rapidly, 50 degrees Celsius, 80 degrees Celsius, 120 degrees Celsius. Under the microscope, the two tardigrades curled into their ton shapes again, the natural defensive response to extreme conditions. At 150 degrees Celsius, most microorganisms would be reduced to lifeless debris. But when the heat cycle ended and the slides cooled, Elena leaned toward the microscope and frowned. Because both specimens were still intact, still in ton form, still apparently waiting, and the radiation scanner had not dropped. In fact, the numbers had climbed higher, which meant something inside those tiny bodies was not just surviving the experiment, it was adapting. And if that adaptation continued, the scientists were about to face a question far more disturbing than any they had asked before. Not can tardigrades survive space, but something much worse. What happens if something inside them starts evolving faster than we can study it? Laboratory sterilization protocols are designed with a comforting assumption. Life has limits. Bacteria die when heated. Viruses degrade under radiation. Even the hardiest microorganisms eventually surrender to extreme pressure, toxic chemicals, or prolonged energy exposure. The exact thresholds vary, but the principle is universal. Life bends, eventually it breaks. That assumption had quietly guided biology for centuries. It was the invisible rulebook behind every containment chamber, every autoclave, every destroy sample instruction written in lab manuals. But on that night in 1974, inside a quiet research facility that most history books never mention, two tardigrades were calmly ignoring the rulebook. After the heat cycle finished, the specimens remained in their curled ton forms. At first, that was not alarming. Cryptobiosis is, after all, a survival strategy designed for precisely this situation. Tardigrades can endure extreme heat for short periods by shutting down metabolic activity and stabilizing their cellular structures. But the real concern appeared on the radiation scanner. The numbers had increased again. Dr. Orlov studied the readout twice before speaking. Check for instrument error. The technician recalibrated the device. Same reading. Checked against a control sample. Normal. Returned to the 2 experimental specimens. Still elevated, in fact, slightly higher than before. From a purely scientific perspective, this was baffling. Radiation exposure normally damages cells. Even tardigrades, miraculous as they are, must repair DNA damage after such stress. The process costs energy. It slows the organism down. But these specimens seem to be doing the opposite. Every stress applied drying, radiation, heat appeared to make them more active once revived. The second climax of the story began when Dr. Orlov made a decision that, in hindsight, many investigators believe he immediately regretted. Rehydrate them. No one argued, though several later admitted they felt uneasy about it. Scientists are trained to push experiments further. Curiosity is both their greatest strength and their most reliable way to cause trouble. A droplet of distilled water touched the first slide. Within seconds, the ton expanded. The creature unfolded. But this time, the movement wasn't fast, it was controlled. The tardigrade stood upright on its tiny legs and remained still for several seconds, as if adjusting to its environment. Under the microscope's magnification, its clawed limbs flexed slowly. Then it began to move, not randomly, directly toward the second specimen. The room filled with quiet murmurs. Both animals approached each other like magnets, finding alignment. When they met in the center of the slide, something happened that none of the researchers had ever observed before. They synchronized. Their legs moved in identical rhythms. Their bodies shifted in the same patterns. Even their turning angles mirrored each other with eerie precision. It looked less like 2 organisms and more like a single system split into 2 bodies. And then the scanner spiked. The radiation reading jumped so sharply that the device emitted a warning tone. Elena instinctively stepped back from the bench. "What did they do?" she asked. No one answered immediately. Because Dr. Orlov was staring into the microscope with a look that witnesses later described as scientific disbelief mixed with genuine fear. "They're not exchanging radiation," he said slowly. "They're amplifying it." The theory formed rapidly. If the tardigrade's protective proteins had mutated under repeated radiation cycles, they might not simply shield DNA anymore. They might store energy temporarily during cryptobiosis, like microscopic biological capacitors. And if 2 altered specimens interacted, that stored energy might transfer or combine. A biological resonance effect. 2 microscopic animals unintentionally acting like a feedback loop, which meant every revival cycle could increase the stored energy further. At that exact moment, the laboratory lights flickered. Once, then again. The room's electrical monitors showed a brief voltage dip across the circuit line connected to the experiment table. Someone laughed nervously. "Old building," one of the assistants said. But Dr. Orlov was no longer watching the electrical panel. He was looking at the slide. The 2 tardigrades had stopped moving. They were facing the same direction now. Toward the edge of the glass. Toward the seam where the slide met the containment chamber. And slowly, very slowly, both creatures began walking in perfect synchronization toward the boundary. Elena leaned down to adjust the focus. "What are they doing?" Dr. Orlov answered without taking his eyes from the microscope. "They're following the trail." The same trail pattern photographed earlier. The one that had appeared inside a sealed dry chamber before any water was added. The one that implied movement during complete cryptobiosis. The 2 specimens reached the glass edge, their claws pressed against the barrier. And then the radiation scanner emitted the loudest spike it had recorded all night. The next line in the archived report is short, just 8 words written in shaky handwriting. Containment glass showing microfracture along specimen contact line. In other words, the barrier between the organisms and the laboratory had just started to crack. And according to the security logs recovered in 2026, that was the exact moment the building's night guard reported seeing something moving across the floor of the observation room. Something so small he almost ignored it. Almost. The night guard's name was Viktor Sokolov, and according to the security files released in 2026, he was not a man prone to imagination. His personnel report described him in the most unexciting terms possible. Punctual, quiet former railway technician, known for drinking tea during night shifts and finishing crossword puzzles before dawn. In other words, exactly the kind of person chosen for security in a research building full of delicate equipment. People like Victor do not file dramatic incident reports, but at 02:17 a.m., he did. The report begins with a sentence that investigators would later quote repeatedly because of how strangely casual it sounded. Observed small movement on laboratory floor near observation room B. At first, Victor assumed it was an insect. Old buildings attract them, especially in quiet facilities where lights stay on through the night. He walked closer with a flashlight, expecting to see a beetle or perhaps a spider. Instead, he saw something slower. A tiny shape crawling across the polished floor tiles. At this point, the report becomes oddly specific. Victor crouched down and held the flashlight closer to the ground. The object was smaller than a grain of rice, pale in color, with what looked like several tiny legs pushing against the surface. He wrote one short line in the margin of the form. "Looks like tiny bear," which would be amusing if it were not scientifically impossible, because tardigrades cannot survive long outside water films or moist environments. On a dry laboratory floor exposed to open air, one should immediately enter cryptobiosis. Movement would stop completely. Yet Victor watched the thing crawl, not fast, but steadily. Across the tile at that exact moment, inside the observation room, Elena and Dr. Orlov were staring at the cracked containment slide. The microfracture along the glass seam had widened slightly. Under magnification, it looked like a hairline canyon. One of the specimens was gone. The silence in the room lasted several seconds before Elena finally whispered the obvious question. Where did it go? The chamber had not been opened. The lid remained sealed. The fracture was microscopic, far too small for a normal hydrated tardigrade to squeeze through. But a dehydrated ton? That was different. In cryptobiosis, tardigrades shrink dramatically. Their bodies compress into compact shapes far smaller than their hydrated forms. Small enough to slip through spaces most scientists would never consider dangerous. Dr. Orlov's voice became very quiet. Search the room. Under normal circumstances, that instruction would sound ridiculous. Looking for a 1 millimeter animal in a laboratory is like searching for a single grain of sand on a beach. But this was no longer a normal circumstance. They began scanning surfaces with flashlights and magnifiers, tables, floors, instrument bases, even lab coats hanging by the door. Nothing. Meanwhile, Victor was still crouched in the hallway, staring at the tiny crawler. He later testified that he nearly crushed it out of reflex. Security guards are trained to eliminate pests in sterile environments, but something stopped him. Perhaps curiosity, perhaps the odd shape. He leaned closer. The creature paused as if sensing the light. Then it did something that Victor would repeat several times during the later investigation. It stood still and curled. Right there on the tile floor, the tiny body contracted into a tight ton shape. Seconds passed. Nothing happened. Victor frowned, lowered the flashlight, and leaned even closer. "Dead bug," he muttered. Then a drop of water fell, not from a pipette, from Victor's own teacup. He had been holding it loosely in his other hand, and a tiny droplet slipped over the rim as he crouched. The droplet landed directly on the ton. What happened next lasted less than 3 seconds. The tiny body expanded instantly, 8 legs unfolded, and the creature launched forward across the tile at a speed Victor later described as faster than any insect that small should move. Startled, he jerked backward and spilled the rest of his tea across the floor. In the observation room, alarms suddenly began sounding. Not radiation alarms, electrical alarms. Several instruments connected to the laboratory circuit board spiked simultaneously, drawing brief bursts of current before shutting down. The building lights flickered again. Elena rushed to the monitor display. "The scanner is picking up movement outside the chamber," she said. Dr. Orlov looked toward the door, and that was when Victor burst into the room. "There's something on the floor," he said breathlessly. "Tiny animal, running." 3 scientists and 1 security guard stood frozen for a moment. Then the radiation scanner chirped again, a moving signal, heading slowly toward the doorway, toward the hallway, toward the rest of the building. The official experiment log ends shortly after that moment. The next surviving document jumps forward several hours and contains only a brief administrative statement. Facility placed under temporary quarantine, biological samples secured. But the most intriguing detail appeared in the documents released in 2026. When investigators reopened the sealed storage boxes, they found something unexpected attached to Victor's original report. A microscope photograph. Taken at 02:31 a.m., it showed a single tardigrade on a glass slide recovered from the hallway floor, completely dry, curled into a ton. And beside it, barely visible under magnification, were 3 sets of microscopic trail marks. Not 1, not 2, 3, which raised the question that still bothers scientists today. If only 2 specimens existed in the experiment chamber, then where did the 3rd one come from? And perhaps more importantly, how long had it already been moving through the building?